Hello, students, and welcome back to the 2020 to 2021 AP Biology school year here at TASM. I hope that you had a good summer, that you got to connect with family, and that you were able to stay safe during this challenging pandemic. This image in the top left was my last big run of the summer. I got really into running, and I just had serenity with this sunrise here over Zeba Beach. And I'm looking forward to being able to teach you this year and to work with you and hopefully to work together. We hopefully get that vaccine and get back into this school. All right, just like at all times, we're going to rise to the challenge here at TASM. Reach out to me if you need help. Reach out to your classmates, and we will get through this together. This video covers Unit 1, the chemistry of life in AP Biology. This is to review your summer assignment, okay? All the images in this presentation come from Khan Academy or OpenStax or from a Google search where the Creative Commons license allowed it. Just as I said about this license, this video is not for profit. Okay, I will make no money off of this video, and nobody else should make any money off of this video. It is solely meant to help students in their quest to master AP Biology. Some of the slides I did get from Jamie Kessel, who ran an AP Biology review session in the spring that was widely attended, and I will put a link to that in the notes on this YouTube video. As you're reviewing this, I want you to remember I did have you read in the Campbell textbook the chapter on the themes of biology. And there's 4 big ideas or 4 really big themes in this course. And they are evolution, energy, information, and interactions. Evolution, providing for the unity and diversity of life. Energy, photosynthesis and cellular respiration and homeostasis. And then information, DNA, RNA, protein, and interactions, how organisms interact together, how molecules interact together, how cells interact together, etc. All right, so you should be able to see them in all aspects of this unit. And I really encourage you to think about evolution. In the past, before AP came out with this curriculum, I always thought evolution first, and it really provides a foundation for it, and we'll talk about that in class. Okay, there are 6 subunits in here: the structure of water, elements of life, biological macromolecules, and nucleic acids. We'll go through all of them. This video is going to focus on content. It doesn't really focus on the scientific practices. We will use tons of them in class. So you can't get by just memorizing the content. If you just memorize this video, it might help you, but it won't be the full thing that's going down in AP Biology, right? You need every AP question combines a scientific practice and some piece of content. So you really need to practice the ways of doing science, and that's what we'll be doing in class. Whoa, what is this picture here? Don't even try to pause the video and read my handwriting. It is terrible. But the reason I put it in here is this is a concept map I made myself as a teacher as I was trying to think about this unit and what I want you to get out of it. I made this concept map by reading the summer assignment I gave you this summer and by looking through the AP curriculum that I also have posted to Google Classroom, what you need to know content-wise. So this kind of lays out for me in my head what the content of this unit is. And I would encourage you to do the same thing afterwards, to look at this video, to look at your notes, to look at the slides, and to try and come up with a one-pager, just one piece of paper where you kind of go through and figure out what's going on in this unit, so that way you don't get lost amongst the trees in the forest. You can see the whole big picture. I had students do this for all the units last year. Last year we were tested on units 1 through 6 for the AP exam, so they had 6 of these one-pagers. You'll have 8 because I believe we'll be tested on units 1 through 8. Okay, let's dive into the content. So first things first, you don't need to know a ton of chemistry for this course. It will help you. It won't hurt you to know some chemistry, but they have split up AP Biology and AP Chemistry. And here at TASM, you are allowed to take AP Biology without having taken chemistry before, as some of you in 10th grade are doing. And it's perfectly fine. And I agree with that for the course. But you'll have to know a little bit about how elements interact together or how they bond together to kind of get what's going on, especially here in these first couple units. So if you are shaky on your chemistry, you can check out my pre-AP Biology Chemistry video that I published at the end of last school year. It's also on my YouTube channel. That'll help you go over this a little bit. But just as a quick refresher, elements are one type of atom. So hydrogen would be an element. And then oxygen would be an element, carbon, nitrogen, sulfur, phosphorus, etc. You can look at a periodic table, and the number at the top is the atomic numbers, the number of protons for that element, and that's what makes it that element. Different numbers of neutrons would be what are called isotopes. So here, if you look up hydrogen, it's going to have one proton, and it's going to have one electron. That one electron orbits the proton. Here are two different models, right? And these are models showing us how we can think about the element hydrogen. So here it's just kind of a cloud around it. The electron's a cloud around that proton. And here it's in more of a defined circle. And this is called a Bohr model. Here is a larger element. This is oxygen. And oxygen is going to have 8 protons and 8 neutrons. So it'll have an atomic mass of 16. And it's going to have 8 electrons circling that nucleus of protons and neutrons. How amazing is it that these negative electrons circle this? It's pretty cool. You should ask Mr. Kelly and Mr. Bywater about that. So on the first level, it's going to have 2 electrons circling it. And then on the second level, it's going to have up to 8 electrons circling it. Here it has, let's count them. 1, 2, and then 1, 2, 3, 4, 5, 6. So 6 in the second level. And one thing I taught my students, and I used to teach physical science, is that 8 is great, the octet rule. So this oxygen is looking for 2 more electrons out here. Now, it's not actually looking. I shouldn't give it human qualities. I shouldn't anthropomorphize it. So it has the ability to bond with 2 more electrons, which we will see it do. So atoms by itself are one thing, but there's an emergent property. And the emergent property is when they bond with another atom, forming a compound or a molecule. They can often do more than what one atom could do by itself. So go back to this slide. Here's hydrogen and oxygen. Well, you know, if 2 hydrogens and 1 oxygen get together, then that's water, and that's really important to life. And that's going to be different than these 2 gases on their own. So that is called an emergent property, where the whole is greater than the sum of the parts. And you're going to really see that with water chemistry coming up. Okay, so let's dive in. The structure of water and hydrogen bonding. So this would be a good point for you to pause the video and see if you can fulfill this learning target here, or this learning objective here, with the knowledge that you gained from the summer assignment. All right, let's see if you got it. So here is a water molecule. And so it's going to have a polar covalent bond. The oxygen is going to be a more electronegative element. It's going to attract electrons more than the hydrogen does. So it's going to have the electrons from the hydrogen around it more often. So the oxygen is going to be more negative. This is a covalent bond. So that means that these are sharing electrons. It is a very strong bond right here. And then the O, or the oxygen, is more slightly negative than the H, or the hydrogens, which are more slightly positive. You can think about it looking kind of like Mickey Mouse. If you're to flip this around, this would be happy ears and a sad mouth. All right. So here is an example of an emergent property from that. Because this water molecule is polar, the oxygen is negative and the hydrogens are positive, the oxygens are going to have a hydrogen bond with the hydrogen of a nearby water molecule. Notice how this is a dashed line right here. These are hydrogen bonds. This hydrogen bond is weaker than the covalent bond within the water molecule. So between this H and this O is a very strong covalent bond that is not going to break right here. But there are weaker bonds in between the oxygen and the hydrogen of another water molecule that are called hydrogen bonds. And these hydrogen bonds are really, really important in biology. They help to hold together our DNA, and they perform a lot of functions. It's actually the strongest type of intermolecular bond. And so inter means between molecules, not intra, which means within. So these covalent bonds would be within the water molecule. These are between them. All right, so what is a hydrogen bond? It's an attraction between a nitrogen, an oxygen, or fluorine. These are all very electronegative elements. It means that they attract electrons a lot. They're you can think of them as stronger than this hydrogen. And so it's an attraction between an NO or F and the hydrogen that's going to be bonded to an NO or F. So think about what that means. Why does that work like that? Well, if the hydrogen's bonded to nitrogen, oxygen, or fluorine, then it's going to tend to be more positive because the nitrogen, oxygen, or fluorine will attract the electrons more often, which is what's going to make it a polar covalent bond. There's some chemistry you can do with electronegativities and the Pauling scale to figure this all out, but you don't need to do that for this course. You just kind of need to see the big picture here. And then this is a really good example right here that Ms. Castle put in here. Here would be H's that will not form hydrogen bonds because the electrons are being shared equally. So between this carbon and this hydrogen, carbon has 4 valence electrons, so it wants to bond with 4 other electrons, or doesn't want, it has the ability to. And so here they're shared evenly. So there's no positive and negative ends. So therefore, there's going to be no way that this is going to do or engage in hydrogen bonding. This will be important with fats and oils and lipids later on. Okay, what is big idea number 4? The interactions of systems. So water is a great example of emergent properties coming from systems. When they interact with hydrogen bonding, they create many of the properties that give rise to life that are really important to us and to living. So pause the video here and see if you can label them out. Okay, hopefully you got cohesion, adhesion, capillary action. Transpiration is kind of where these all combine together. Transpiration is water going through plants from their roots all the way out through their stomata on their leaves. The ability of water to store a lot of heat. Think about the ocean or think about your sweat. And it can even help to cool you down as you sweat. The water molecules with the most energy will be leaving you, so it'll help to cool you down. Water is less dense as a solid than as a liquid. Why is this important, right? Well, it's going to make ice float. Think about it if oceans or lakes froze from the bottom up. That'd be a real bad thing for fish. And then water is a universal solvent. Because water is polar, it has positive and negative ends, it's going to be able to pull apart many large groups of molecules. Think about a cube of sugar. And so it can pull apart all the different sugar molecules so that they're more spread out or diffused. And so here is just a little bit of a picture of transpiration. And so because of cohesion and adhesion, because the water molecules stick together and they're able to stick to the sides of the plant xylem vessel here, it is able to get to the stomata or to the hole in the leaf, and then it will evaporate out, okay? And in here is ice and water. So when water freezes, the hydrogen bonds lock in place, and it forms a lattice type structure, and that's going to have more spacing here in between the different water molecules. So look at this: 1, 2, 3, 4, 5, 6 water molecules. And here's 8. Notice how you can fit more in the same space right here. So when it's water, they're constantly slipping by each other, etc. So density is mass divided by volume. So here, if the volume is larger, then the density is less. And that's what lets ice float. All right, and here's just a couple other pictures of it. Okay, the elements of life. This was a really easy section. What's required for life? All right, well, hopefully you got sponge. Right? Sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen. Of course, there's other stuff required for life because some of them will be cofactors for enzymes, etc. But we can mainly focus on these to begin with. So can you tell me all the elements that go in carbohydrates? Hopefully you got carbon, hydrogen, and oxygen. How about the elements that go in lipids? Same thing. So this is interesting, right? The arrangement of carbons, hydrogens, and oxygens in carbohydrates is different than the arrangement of carbons, hydrogens, and oxygens and lipids. So a different structure leads to a different function of the biological macromolecule. All right, lipids, phospholipids do have phosphorus in them, just so that you can know that's a little trick there. But most of them are just carbon, hydrogen, and oxygen. How about proteins? What goes in there? Well, you're starting to see a trend, right? Carbon, hydrogen, and oxygen, as well as nitrogen. And so that's going to be in that amino group. All right, and then there's some of the R groups will have sulfur in them, cysteine, for example. And then how about nucleic acids? They're going to have carbon, hydrogen, oxygen, nitrogen, and phosphorus. And so this was really important in the experiments to discover the structure and function of DNA. And so they used that knowledge to figure out what was the basis for genetic material. And we'll go over that in Unit 6. Okay, so can you talk about monomers and how those monomers get connected together to make polymers or biological macromolecules? Okay, hopefully you got something along the lines of this. A polymer, poly means many, are large molecules made up of many similar molecular building blocks. So my kids love Legos, and so I see them making polymers of Legos all the time. The monomers for carbohydrates are saccharides. So if you put a bunch of them together, they can be disaccharides and they become polysaccharides. The monomers or proteins are amino acids. When you put a bunch of them together, it's called a polypeptide chain or a protein. The monomers of lipids or fatty acids. Lipids don't really have a monomer, but if you wanted to say one, you could say fatty acids, and then they're connected to a glycerol. And the nucleic acids are nucleotides. A sugar, a phosphate, and a nitrogenous base, and then they get linked together to make your DNA. All right, so let's talk a little bit about making them and breaking them, right? You're going to be constantly making them in your body to make proteins. When you're looking at me, you're mostly looking at proteins that I have made. And then when I eat proteins, I'm going to be breaking them down so that I can use those amino acids to make new proteins. So what is that called? It is called dehydration, synthesis, and hydrolysis. I've had students in the past who have struggled with these, and I tell them to just think about what the word says. Dehydration, water leaving, and synthesis to come together. So if you take out a water molecule, when you're putting together these two monomers, you'll get a polymer. If you insert a water molecule, hydrowater, and lysis means to break. If we insert a water molecule, we can break this macromolecule or this polymer down into monomers. We'll see some pictures of it. So here would be glucose and glucose coming together. Here is what's called a hydroxyl group. It's going to take this OH and this H, and they're going to be spit out or released, and it's going to release a water molecule, H2O. This is catalyzed by enzymes. Right? And then it will be bonded on this oxygen. It's called a glycosidic linkage. Here would be hydrolysis, water breaking apart. All right, and speaking of water, I'm going to take a sip of one now. I'm back. So here's maltose, a disaccharide. So if you use an enzyme, maltase, to break this down, it's going to insert the water molecule, and look where it does it, right on this oxygen. It's bridging them together. It's going to insert an H here to make this a hydroxyl group, and this H here, also a hydroxyl group. Don't worry about that word. And so it's going to insert the water molecule to help break these apart into their monomers. So hydrolysis, water inserted in to break apart. Dehydration, spitting out a water molecule to bring together. This is also called a condensation reaction. If you think about condensation on a glass, you see water there. Okay, let's look at some of the properties of monomers and types of bonds that connect the monomers into biological macromolecules. I'm going to combine that with structure and function. All right, we already did this, so I had this slide in here twice. Sorry about that, but maybe it is nice to look at. Okay, let's talk about carbohydrates first. The only thing my kids will eat. My goodness, they love French fries. So carbohydrates are carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio as a monomer. As a polymer, it'll be in a 1 to 2 to 1 ratio as well, except for the water molecules that have been lost by dehydration synthesis. So this is kind of a fun one. You can pause the video here and kind of write out these 6 glucoses together and see if you get the same thing here. Carbohydrates often end in the letters OSE. So if you see something like glucose, maltose, sucrose, fructose, you know that they're a carbohydrate. And they're made through the process of photosynthesis, which we will study in depth and in detail later on in the course. Their monomers are called monosaccharides. Pretty easy with the same letters there. Simple sugars are one sugar. Some examples would be glucose and fructose. Your body will break things down into its monomers before you can begin cellular respiration on these sugars, where your mitochondria breaks apart those sugars to release energy in the form of ATP. Polymers. So you have disaccharides, 2 monosaccharides joined together on that glycosidic linkage. Some examples would be maltose and sucrose. Polysaccharides are many of them put together, maybe hundreds of thousands of them. And this is interesting, right? It doesn't always have to be energy storage, such as starch or glycogen. It could also be structural. There are structural carbohydrates, including cell walls, including chitin in the exoskeleton of spiders and crustaceans, and in the cell walls of bacteria. Here's just some examples of how the structure of a molecule can lead to its function. Just different ways that the carbohydrates are arranged when the monomers are put together give you different molecules. You do not need to memorize that, not at all. They will give you context clues on the AP exam, and this is a little bit beyond or outside the course. Okay, here are fats, and so fats are going to have this fatty acid tail. This is going to be non-polar. These hydrogens are bonded to a carbon. They are not bonded to an electronegative oxygen, nitrogen, or fluorine, which would give you a polar molecule. So in other words, this isn't charged. It's just this long chain of carbons and hydrogens, and it's going to be non-polar, which means that it's going to repel water. It's going to bond with this glycerol molecule, and that bond is called an ester linkage, where it spits out a water molecule. You might be thinking to yourself, well, isn't that just dehydration synthesis? And that is true. And we use the name ester linkage just so we know that we're talking about making a fat here, just like a glycosidic linkage was for carbohydrates. And so let's see what I have over here. It's 3,8 fatty acids form a fat. The alcohol group, or the hydroxyl, is removed, and a carboxylic acid group with the elimination of water is formed. And so these saturated fats, this is not important for our course right here, but what is important are structure leading to function. So let's look right here. Let's see if we can see it, or it might be on the next slide. But here, where they're all single bonded carbons, there's going to be no kinks, and this is going to be a saturated fat. This is going to be solid at room temperature, something like butter. But here, if it has a double bond, then it's going to have a kink, and it's going to be liquid at room temperature. And that'll be important because when we make phospholipids, you need a combination of saturated and unsaturated fats to have the right fluidity of the cell membrane. So unsaturated fats have a double bond in them somewhere. It's going to give a kink to that part of the molecule, and it's going to make it liquidy at room temperature. An example would be olive oil. Here is an animated GIF to show you the dehydration synthesis happening. Here you can see a fat being broken apart. So I have been running really hard this summer to try and break apart the fats on my body and to release them as carbon dioxide and water. Here is a lipid. It's a great example of structure determining function. So here are those fatty acid tails, but instead of having 3 of them, the third part is going to bond with a phosphate. And this phosphate is going to give this part of the molecule polarity. It's going to love the water. It's going to have a charge, and it's going to say, hey, water, you are great. We're going to hang out with you. Whereas the tails are non-polar, and they're not going to like water. They're going to try and get away from the charges of water. And so naturally, they will form lipid bilayers, where if you put enough of these in water, it won't take any energy. They'll just naturally form this. And so you'll have the non-polar tails pointing to the inside on both sides and the polar heads pointing towards the water. So just remember, polar likes water. These bilayers are really important. They're going to become cell membranes, and then there's going to be channels through them. But this non-polar interior will repel any charged molecules. Okay, we're rolling along. If you're still with me, you're doing great. All right, one more lipid that we'll go over in more detail later on are steroids. And these count as lipids because they're hydrophobic. They are non-polar. I've been using those terms a lot in the same. Hydrophobic means water-fearing. Non-polar means it doesn't have a charge. It doesn't like water. And so they're going to not dissolve in water. They're going to have a different structure. And then they're going to have little molecular tags on them that are going to give them different functions. So look right here. You can see testosterone versus estrogen. They look very similar, but they just have slightly different molecular tags in different parts. Okay, proteins. Probably the most important biological macromolecule I guess DNA and RNA are really important as well. But it's really important to understand proteins. All right, and it's a lot of what we'll work on in this course. So proteins are a bunch of amino acids put together into a compact shape. Those shapes determine the function of the protein. So they're really, really important. They do everything, right? They help to promote chemical reactions as enzymes. They help to make your muscles. They help to make your hair. You'll notice I have a lot more hair because I haven't been to the barber. And they're made of carbon, hydrogen, oxygen, and nitrogen. And as I said before, sometimes the R group will have sulfur. So it's really important to understand the 4 levels of protein structure. The primary structure are just the order of the amino acids. And there's 20 different amino acids. We get them from the different foods we eat, and you synthesize some of them in your body. They're all exactly the same except for the R group. So the amino group and the carboxyl group are the exact same, but the R group is different for all 20 of them. And because of that, they'll have different tendencies. But this backbone is the same. So you can bond them all together, and then the R groups will determine which way it folds or shapes. And so whenever we're back together, we'll do a protein party where you'll all have different R groups, and you can wear it as a hat, and then you have to move around, etc. And so those who are polar will go towards water. Those who are non-polar won't. All right, and here's just an example of the different R groups and what they can look like. You'll see that this is chemistry shorthand here. They're put together by what's called a peptide bond, another name for a dehydration synthesis reaction. Remember dehydration, taking out a water molecule, synthesis, putting together the 2 monomers. So here the amino acids are put together into a polypeptide chain. So proteins will be thousands of these long put together. Okay, they're always added on the carboxyl end. The carboxyl end is the COOH. So they do have some directionality. So it'll be added on right here as they're forming. You might remember from 9th grade biology or wherever you took intro biology, the process of protein formation where DNA codes for RNA. The RNA leaves the nucleus and goes to a ribosome where it's going to be read by the ribosome, and tRNAs are going to bring these correct amino acids together, and they'll help to catalyze this reaction right here. The primary structure is just the order of these amino acids. So for example, tryptophan, cysteine, valine, glutamic acid, etc. It's just the order in which they're put. That cannot be changed. They cannot be changed from the order they're put in. Now you might have like a DNA mutation or something that changes it. But what I mean is, as you're making a protein, you don't change the order of them. The secondary structure is going to be, as the protein is being put together, there will be hydrogen bonding between the oxygen atoms and the hydrogen atoms in the backbones of the amino acids. And it gets a little complicated, but they'll either form alpha helices or beta pleated sheets. And so they'll either form little squiggles or beta sheets here. The actual turns and why it does this and what catalyzes that happening is beyond the scope of AP biology, but it's interesting. This is the secondary structure. Here is something really important. This is the tertiary or the third structure level. And these are where the R groups start to interact, right? And so there'll be some R groups that really want to bond together. So they'll form what's called a disulfide bridge. So if they each have a sulfur in them, they will form a bond together. Here, they might be near each other and want to form a hydrogen bond. Notice how this is an OH right here. So this H is going to be slightly positive, and it's going to form a hydrogen bond with this oxygen hanging off of this carbonyl here, if you've done some chemistry. You could have hydrophobic interactions. So that means these two R groups, both of them don't like water. So they're both going to stick towards the inside of the protein. So what is the big picture here? In tertiary structure, the R groups, or what is different for each amino acid, will start to have their own interactions. And those interactions, they'll be facilitated by what's called chaperone proteins, but they'll help to give the protein shape. And so we'll study in class a disease called sickle cell anemia, where one of the amino acids is changed. And so there's a mutation in DNA, and it can be passed on. And if you get, a copy from both mom and dad, then your red blood cells, when they're made, they will have this mutation in it where there'll be a substitution error for an amino acid. And so it'll change it from something that is, it'll change the polarity of it. And so the actual ones are glutamic acid being changed to valine. All right, and in quaternary structure, or the fourth level of protein folding, so here's a good picture of it, right? Primary is the amino acids put together. Secondary is the alpha helices or the beta pleated sheets. Tertiary are those R groups interacting. And in quaternary structure is multiple proteins coming together. For example, making hemoglobin. It's going to have 4 proteins come together to give it the correct shape. All right, and so this is important. The secondary, tertiary, and quaternary structure can all be denatured or messed with. And so if you have extreme changes in temperature or pH, which is the hydrogen ion concentration in a solution, then it can change the interactions between these R groups. And when you denature the protein, then it's going to change the function of the protein. And it usually will not work as well. Okay, we're almost at the end. If you've made it here, you're doing great. I hope you found this helpful. DNA and RNA and nucleic acids. Okay, we're going to go over this a ton in Unit 6, but this is a good intro here. So here will be a nucleotide, a phosphate, a sugar, and a nitrogenous base. This backbone, sugar-phosphate backbone, is the same for each nucleotide, but the nitrogenous base is different. It can be adenine, guanine, thymine, or cytosine, or uracil in the case of RNA. All right, and so adenine and thymine are always going to pair together, and guanine and cytosine are always going to pair together. And let's see why. The reason why is that A and G, or adenine and guanine, are purines. They're going to be slightly bigger in shape. And then here you can see them. And then here are the pyrimidines, cytosine, thymine, and uracil. So in order for a molecule to work, it's always got to have a purine matched to pyrimidine in order for DNA to form the correct double helix that it can be stored in the cell. Here's a way I remember A's go with T's: ashtray, garbage can. You should put ashtrays in a garbage can. You shouldn't smoke or Juul, it'll kill you. That's just something I remember from my 9th grade biology teacher. You might have come up with a different one, and that's great as well. But A's go with T's and G's goes with C's. You probably read in a book that there's some directionality in DNA. So There's a 5' end and a 3' end. That's just talking about the location of carbons on the nucleotides. This is going to be important because all you really need to remember is that nucleotides are always added to the 3' end, because that's where a free hydroxyl group is, or a free OH, where they can do the dehydration synthesis reaction. All right, and a little bit of the difference between DNA and RNA. DNA has a different sugar. RNA uses uracil, whereas DNA uses thymine. And DNA is double-stranded, whereas RNA is single-stranded. RNA can leave the nucleus, whereas DNA cannot. And RNA is subject to more mutations when being synthesized, which is one of the reasons that RNA viruses are harder to or mutate more quickly. So I hope you've enjoyed this video. One thing I realized is I did not go over acids and bases as much in this. Just think about them as H+ concentration. We'll go over that in class. And then here you can pause and look at a few of these sample free response questions. All right, students, take care. And I hope that you enjoyed the summer assignment and get excited as we build upon this unit to try and solve some really important questions in biology, such as cancer and other diseases. Have a good one.